move it. Let's see.
Dr. Bedair, also we have another guest. Dr. Yansal is here right now today.
Hello, Dr. Yansal. Hi, Professor.
Hello.
Nice to meet you.
Nice to meet you.
Bilge, you can start.
Okay. Hello, dear all. Welcome to our first Biotalks event that organized by Hacettepe Food Student Club. First of all, I want to introduce myself. My name is Bilge Yılmaz, and I am a fourth grade student at Hacettepe University Food Engineering Department. I am also a head member of Hacettepe Food Student Club Biotechnology branch. I am glad and pleased to be here with you today. Dear guests, if you have any question after the speech, you can send from chat to host and co-host. We can move forward your questions to the speakers. We have very important speakers among us. As Hacettepe Food Student Club, we are very honored and we want a special thanks to Dr. Mohamed Bedair and Professor Dr. Yahya Şems Yönsel. Also, I want to thank to our dear teacher, Associate Professor Remziye Yılmaz. She was always with us and always supported us during planning of this event. Now, I want to give my word to our teacher.
Oh, thank you. Thank you so much, Bilge. And I would like to thank for our guest today. The time is very important for us and also for them. They are so important people and we are a little bit excited to host them. Thank you so much. And Dr. Bedair, actually, he's coming from the Bayer Crop Science. He's going to work at the Bayer Crop Science right now, and he's responsibility on compositional biology and regulatory sciences right now. And thank you so much for your time, Dr. Bedair, and for your presentation from now. Please share your information with us.
Thank you very much. And I am very pleased to be here. Thank you for the invitation and for the great opportunity. I guess that pandemic made us all comfortable having virtual meetings, but I certainly look for the day that I can come and visit Turkey and, you know, have this seminar face to face with all of you.
Yeah, please.
Sure, you know, hopefully that will come definitely soon.
Yeah.
So again, my name is Mohamed Bedair. I lead the composition and biology team in regulatory sciences in Bayer Crop Science. I am stationed in St. Louis. This is the headquarters for the crop science in the United States. And my talk today is mainly two talks kind of like put together, but they are related. So the first part will focus on safety assessment of genetically modified crops with focus on crop composition. What is the current workflow? What is the current procedures and guidelines that we use to do it? And then in the second talk, or in the second part, I will give a brief overview on metabolomics and the call for using metabolomics in risk assessment, and what are the challenges that we see that is still needs to be overcome to have it as a useful tool for risk assessment. If you have any questions, if you can write them in the chat, or if you want to wait until the end, but please don't forget it. Just write it in the chat, and we can address it at the end, definitely. So I think when I'm speaking to this group, there is no need to emphasize the innovation in agriculture is key to allow us to grow more with less. This is the only way that we can feed a growing planet, a growing population on a limited arable land, or even decreasing arable land. This is actual plot that is a few miles away from St. Louis, in a farm, few miles away from St. Louis. So it's not a constructed picture, but these different plots were planted using the technologies in each year that you can see. So you can see in this big plot here, the 1940, you had to grow 1,400 square meters, to get 250 grams of corn yield. And with the great technology of hybrid breeding and agronomical innovations, By the 80s, that only was lowered by almost 1,000 meters square. And then you can see the innovation later, and in the future, that's the only way that we can illustrate to visitors that innovation in agriculture is key for us to still keep feeding a growing planet with the same resources that we have, if not even decreasing resources. In the heart of this technology comes genetically modified crops. This is a meta-analysis that was published a few years ago that shows the benefit. You can see the increase, like almost 20% increase in yield of using GM crops. And the increasing yield comes mainly from decreasing the pressure from weeds and decreasing all of the pest pressure. And as you can see, when you decrease the pest pressure, you decrease the use of pesticide quantities that will benefit the cost, will lower the cost of using pesticide by almost 40%. This will increase the profit that a farmer will reap for his production, even though if he pays more for the production, and mainly that comes from the more expensive genetically modified seeds, but the gain overall is substantial, almost 70% increase in farmers' profit. Genetically modified crops are more expensive because they, and I'll talk about that in the next slide, but in biotechnology, you would think, genetically modified crops as an extension of breeding. When you have a two-line for breeding, for example, you have two, corn lines, and you have traits in one that you need to breed into the other. You can breed them traditionally. If you have a trait or a gene that in an organism that is not, outside of your host plant, you don't want to get from another plant or from a bacterial. Plant biotechnology allow you to do that. So you have the desired gene and you enter it into the host genome, and then you'll have one gene for your gene with the GMO, only inserted in it. And I will, talk more a little bit on that in the end. But as I said, developing a biotech product takes an average of 13 years and well over $130 million. The pipeline development resembles that of pharmaceutics. So we have a discovery phase where gene of interest and traits are identified, usually through high throughput screening, model plans. And then when you have identified the gene of interest or the trait of interest, you advance them to phase 1. When you start doing crop transformation, testing on small plots to show proof of concept and to gene optimize it. Again, advanced events, the successful events, the efficacy events get advanced to phase 2, where they are tested on a much larger scale, multi-trial, so extensively tested, and also large scale transformation, and you start generating some pre-regulatory data. From the beginning, from discovery till the end of phase 2, you can start with thousands of events and genes to test. Ideally, by the end of phase 2, you will have only one event. That's your optimized and most efficacious event that you hand to regulatory. So I'm in regulatory, and we call this a handover meeting, and it's usually celebratory when we get the product from biotechnology department to regulatory department. This is inflection point here. Now we take that event, we go to the field again, we plant it, and then we start all the regulatory data generations in phase 3. Also, breeding takes the trait and start integrating it in other germplasm. So that's why you can see now the number of events will increase, because, or not events, the number of traded products will increase, because as you expect, we don't sell, we sell different germplasm for different geographies, maturity range. Corn sold in North America is different than the South America. So we have to integrate that trait into those elite germplasm that we are optimized for yield and for insect resistance and for many other agronomical breeding technologies. By the end of phase 3, we have finished our safety studies. We form a dossier and we submit it for regulatory agencies for review and for approval. That is in phase 4. And at the same time, commercial sites start building up seed, bulk up, pre-marketing, and product development. So this whole, takes over 10 years, you most likely no one will, see it from all the way to the other groups, and people, handle it at different part of the product lifetime. I want to emphasize that trait selection and breeding into elite germplasm highly minimize the unintended effect, because as you would expect, when we transform, when we generated, a genetically modified, you enter a transgene into the genome. You don't know where it's going to land, so it can have an off type. It can affect an endogenous gene. It can have multiple copies. So it sure will have an unintended effect or off types. But this process, this is an example that was published again a few years ago. For this example, you start for 5,000 transformation, and usually it's much larger than that. And then you start screening one by one. So you screen how many copies. I need only one copy. Do I have the backbone of the ddNA is there or not? I don't want that. Linkage assay, so we have markers to select the one that has the trait. You want the markers and you want the trait itself away from each other, so you can segregate them later on, because the actual event that you want, finalized or optimized, you don't want to have markers for them. You don't want to have any unnecessary genetic, elements there. And then you screen for, efficacy, you screen for expression, protein, field trials. All this process will take you down to this is the best event that we have. Usually this is, lower and minimize that unintended effect a lot. And now that's not the one that we sell to farmers. Again, we take this and then we incorporate it into commercial breeding with elite germplasm. After multiple back crossings, more than 99% of the genome comes from the elite germplasm, and less than 1% of the genome of that product that we sell to farmers will be coming from that transgenic effect. This, I just wanted to highlight it, because one of the great calls for using profiling and using large data is that the criticism that we don't see unintended effect using our traditional approach. When we measure 80 to 100 component and we don't see any unintended effect, it's not because it's not there, but it's because it's there, but we're not seeing it. But this process, if you understand how it is developed in industry and in biotech, that process really highly minimize all the unintended effect to a minimum. Because again, whatever you sell will have less than 1% of the traded, and it has extensively been tested both in the lab and in field. So this is an example of the regulatory studies that we submit to. This is kind of like an FDA example. You have a section on description. What is the host plant that you are transforming? What is the donor? Where did you get that transgene from? History of safe use for both. Then you have a section, of course, to characterize the genetic modification. Description of the vector, the transgene delivery process, characterization of the introduced DNA, sequencing, number of copies, characterization of the insertion site. Where is the flanks of that gene? Is it in an area or that is transcribing genes or not? It's not an open reading frames, or it's safe. It did not have any effect on any endogenous genes, and generational stability as well. You want to test that this transgene is stable for 3 or 4 generations, and it segregates normally. The next chapter would be, what is this transgene producing? So if it's producing a protein, what is the structure of the protein? What is the amino acid sequence of it? What is the mode of action? Specificity is a very important thing. If you are having a herbicide tolerant gene, then will give you a protein or an enzyme that will detoxify a herbicide, you want to prove that it's specific to that herbicide alone, and it's not have a broad activity against non-specific substrates. You want to prove that it does not interact with the endogenous metabolism of the crop or of the plant that you introduced it in. Toxicity of the protein, and that's done by animal feedings for protein acute tox and tox studies, and of course allergenicity as well. You want to prove that that protein that is entered is not an allergen. It's not going to induce allergenicity, And it's not going to affect the allergenicity of known allergens. Like in soybean, for example, there are known allergens in soybean. You want to prove that any transgene that you're going to transform a soybean product does not affect the endogenous allergens there. And actually, allergens is tested with bioinformatically way in the beginning of the pipeline from discovery, because you don't want to spend all this money and all this time developing something, and then you come to phase 2 or regulatory, where you have spent years and millions of dollars and resources, and you find a flag. So regulatory actually, is involved through the whole lifetime of the product. If there's any red flags or anything that will affect our submission, affect the safety of the product, it is discontinued way from the beginning. The third or the fourth bucket of studies, we call them comparative assessment. So we take that event, we send it to the field, they grow a multi-trial field study, and then we have all the phenotypic and agronomic data, plant height, flowering time, maturity, all the other endpoints that you would collect from a plant trial, environmental interaction, non-target organism, and then the compositional assessment. And this is what I'm going to focus my talk for the first part later on. And then nutritional studies as well. So we do broiler feeding studies, 90-day rat feeding studies. We incorporate the whole material into diet that is fed to broiler, to 90-day rat feeding studies, and to catfish as well, in case it escaped to an aquatic system. So we want to have to test it on an aquatic organism. So that's kind of like an overview, and then each country and each region have specific. So we don't have one regulatory dossier for everybody. EFSA has regulations that they require something more than maybe USDA, maybe than the Asia Pacific. So it is a complex process to register all the product through multiple regulatory agencies around the world. So why do we conduct compositional analysis? The compositional assessment provides data to evaluate whether the crop variety that you are developing is as nutritious as and as safe as your conventional counterparts. We use guidelines that is published, and harmonized throughout internationally, like the Codis Alimentarius, excuse me, Codis Alimentarius, OECD, EFSA. These are, many agencies have their guidelines that we follow. I wanted to just highlight 3 of those main guidelines that we follow. Is that we have to compare the test, and when I refer to test in my presentation, I mean a GM crop, genetically modified crop, to a conventional counterpart, a non-GM counterpart that is grown and harvested under the same conditions. So we have to test them side by side. They grow in the same field, and we harvest them at the same conditions. We would like to assess the components that are important for safety and nutrition, that we want to make sure that they did not change to have any effect on health consequences, either for animal feed or for human consumption. So identify what are the components that are important for nutrition, important for safety, test them, and make sure that they have not been altered in any way, form that can affect safety. And then natural variability is a component level, is critical to understand any statistical differences. When you run a composition study, regardless of whether you're going to do targeted assay or omics or whatever, you will always find statistical differences. Plant interact with environment, interact with soil using composition, using metabolites. This is the language that the plant interact with. I guarantee you, if you take seed bought from a soybean from the top of the plant and bottom of the plant with a high power experiment, you'll find differences. So differences when you have a high power statistical trial or a high power field trial, and you find it's a statistical difference, that does not necessarily indicate that there is a safety issue. We have to assess those differences in light of the natural variability. What are the natural variability for the convention crops that have, a history of safe use? Are we within that range? Are we out? So that's the only way that you can have a biological interpretation of the data. So the organization that identified those important components to measure is the OECD consensus documents. There's a document for every genetically modified organism. This is an example from the corn, for example. Actually, the OECD is right now revising that because it's a bit outdated, and they had actually meeting this week in discussing the new document that will come up for maize. So at the end of the document, you have a table like suggested nutritions or nutritional and compositional parameters to be analyzed in maize matrix for human food use. And then it will highlight if you're going to do kernels or grain, you have to measure all this. You have to measure approximate, you have to measure minerals, vitamins, amino acids, fatty acids, phytic acid as an anti-nutrient, raffinose as an anti-nutrient, furfural as well, and then ferulic acid and paraquinaric acid as an important secondary metabolites. So the list will kind of like consist of the key nutrients, key anti-nutrients for a crop, as well as any important metabolites for such crop. For example, canola, you will find glucosinolates, isoflavones in soybean. So each document will identify that. And then there's another table for feed. So that's the items important for human food use. There will be another table that discuss what is the important one to do for feed. For feed, you can do forage because animals can feed on the forage, as well as the grain. Mainly for human food, it would be the processed grain. So whatever flours or any processed from either soybean or from corn or from canola as oil or cotton oil and so forth. So the focus again is on what is important for the commercial use of this crop and how can we assess it in an efficient way. The way we do this is we use official analytical methods. So we standardized method, we use method from the AOAC, ISO, all these are standardized organizations that validate the method and run a ring trial, take the method, run it in multiple labs and see that they get the same results. They publish your parameters, they publish all the what should be the reproducibility, what should be your limit of detection, limit of quantitation, linearity. This makes it a lot easier for multiple labs to use it with confidence that they are using a standardized and validated method, reproducible, and then absolute quantitation is very important. If you want to see one metabolite or one component is higher than the other, you want to know with an absolute quantitation what it is, how much it is, not relative. And all the work to be, by law, for example, in the United States, the EPA, we have to generate this data under GLP. So any work that we do in regulatory that will be included in regulatory submissions, like I showed earlier, has to be done under GLP. That's by law. Because it will, again, provide the regulatory agencies that we have all the quality and the integrity of the data submitted, and then they can go and reproduce it and follow step by step how everything was done. So it's a great quality system that is required in regulatory studies. So as I said, when we get the event from the biotech department, we send it to the field. We usually do a multi-location trial, multiple locations. We pick regions that are represent the growth of the crop, but not in one region. So we want to have, from the east coast or the east part of that region to the west part, north and south, according to what maturity range is going to be included in, and also representative geographical area of where this crop is going to be planted. Experimental design is extremely important because, again, the variability in the data from composition, from ag pheno, all this has to be really addressed earlier on with the right experimental design. We usually use a randomized complete block design. We send the samples to a third party. When we collect the samples are sent to a third party for analysis. usually a CRO that are also under GLP and is certified by the EPA and all the regulatory agencies. And then we get the data back to Bayer. We do the statistical analysis. We do combined site analysis. We don't do side by side. Sometimes we do it if if somebody regulatory agencies will ask for it, but it's usually done with combined site analysis using a linear mixed model, statistical model, and we have a compare test versus control at a p-value of 0.05. Excuse me. All the chain of custody for all the samples has to be maintained through the entire process. So we know with barcoded and with chain of custody from where the sample, from which field went to the sample management lab we prepared the sample, we send it to the CRO, they did the analysis, they send us back the results. All this have to be well documented with a very clear chain of custody, and all this is also included in our trial submission. So when we get the results after the statistical analysis, we look for the difference between test and control. If it is within, it's not statistically different, then we have an implication that there's no food and feed safety implications. If yes, we see statistical difference, then we see what is the test mean and range. Is it within the range of the natural variability on a crop or not? If it is, then again, we conclude that there is no safety implication, because even though there is a statistical difference, that range of the data is within the known natural variability for conventional crop that has a history of safe use. Humans have been using this for thousands of years and with no any health implications. If it is outside the natural variability, then yes, we have to do further assessment. And from composition point of view, the only time we reach this point is then when we actually targeting a compositional component, like for example, developing a high oleic acid soybean or omega-3. So if you are targeting a specific metabolite or a specific component in a nutritional component in a crop, and you're increasing it, that will probably be outside the level. And then at that case, you have to test it. You have to test its toxicity and nutritional value. And so that was the only one when the change is intended, but not an unintended effect. So we have not seen any unintended effect in composition so far in our analysis. When I say natural variability, where's this natural variability comes from? It comes from two main sources: field and growing conditions. As I said, soil, temperature, precipitation, all the growing conditions for a crop, and as well as genetic backgrounds. Different plants. If you have one variety, just one plant variety, and you plant it into multiple locations, you will see differences based on the different locations. And if you have 5 different varieties and you plant them into one location, you will still see differences based on the genetic. And this variance analysis can tell you also that the highest variance that comes from this trial was from the site and was from the genetic background. Trade and trade by genetic background, site by trade, by entry, did not have any residual, or the variance there is much less than you can see between the genetic and the environmental growth. This just an example that you would see from a soy that tell you there is a 4% difference in protein in a 50-acre field. 4% in protein, in total protein in soybean content is a lot, is large. So this contour map will show you that where is it? Even in a 50-acre, you would see differences between crop for just one crop there. How do we get the information about the natural variability? The Agriculture Food System Institute have a crop composition database. It used to be called the ELSI before, but last year they changed their name and affiliation. They are now standalone. They are no longer part of ELSI. They are called the Agricultural Food System Institute, and it is a great database. I'm not sure if you are aware of it or you access it or not. But if not, this is the URL for it. This is the last publication from 2016. Right now, it's on version 8. Was released in last October. Has over 1 million data points there, over 218 component, only conventional. There's no GM data there, so it's only conventional varieties. So it gives a range of what is each of those 218 component in a multiple tissue, different backgrounds, many environments. There are 10 crops, or 10 plants, 10 crops in the database. But as you would expect, corn and soybean has the largest sets of data. This is a growing, again, database that is growing with, we recently added apple, we recently added sugar beet, and rice is also a new addition to the database. Majority of the data, again, comes from the United States, because it's a region that do most of the research for genetically modified crops. But there are data coming from South America, from Europe, as well as Asia. But again, currently still the USA is the largest geographical representation in the database. All the data sent there has to be, again, validated, done with the chain of custody of the sample analyzed within a year. So there is a lot of criteria that controls what is the quality of the data anybody would submit to this database. And there is a review, there's an outlier analysis. There's a lot just to ensure that the data there is as accurate and as representative of the actual natural variability that we can. So, using all these workflow, we have been using genetically modified crops for over 25 years of safe use. There are, since 1996, there's in more than 40 countries, including the EU counted as one. There are thousands of food approvals. There are countries that approve cultivation and import, like South America, for example. There are countries that approve imports only, like Turkey and the EU, with maybe exception of Mon 810 that is cultivated in Spain. There are 28 crops, and I'm apologize again. This is an outdated statistics. It's like 40 years old, so I'm sure these numbers are outdated as well. But it has been used for over quarter of a century without having really safety implications from food or feed. And again, I would like just to emphasize that the commercialized GM crops have equivalent composition compared to the controls, except when the intended GM trade is to improve nutritional composition, like an omega-3 fatty acid or any other high protein product, or if we are targeting that. Other than that, we always see it in the and that also is reflected in animal consumption. When you see this paper again, a few years ago, but it did a meta-analysis of all the animal feeding that was done in the United States since the introduction and even before the introduction of GM crop. This figure, for example, shows you the average milk yield from 1983 to 2011. You would expect that GM was introduced to the market in the late 1990s, and throughout the 2000s until 2011, there is no perturbation in the regression analysis of milk yield. So the regression line showed no perturbation, showed no increase or no decrease. That means that there's no effect from incorporating GM on the on animal production. And this is mainly, the actual life that the dairy cattle will feed on. I would say maybe 90% or 95% of their diet will comes from genetically modified soy and corn. The other lines, this is the post-mortem line, and this is the somatic cell count. Again, you don't see any unintended effect, or you don't see any off effect from incorporating this. For broilers, there have been 94 billion generations of broilers raised and fed in the United States from 2000 in 11 years, 94 billion. So multiple generations, billions of generations of of broilers that were fed on that. And again, if you're not familiar with this article, it's very useful, provide a lot of data on the use of GM crops. So I'm just wanted to highlight that it is safe, has been extended, tested extensively, and the current approach that we are using adequately, represented. So now let's move to the second part. In this second part, I'll just give a quick overview on metabolomics and the call for using and metabolomics in safety assessment. Metabolomics is another profiling technique like genomics, transcriptomics, proteomics. They are used to provide a global analysis of a biological system. Instead of looking specifically for a pathway or specifically for a metabolite, you just want to have an overview of what all is going on. For genomics and transcriptomics, because you're dealing with 4 nucleotides, it's easier to sequence. So in this case, you have a high sequencing rate. You can have a high coverage of the DNA or high coverage of the RNA in a system, because it's easy to sequence. And you have just one machine, next generation sequencing, or whatever technology is there. But they are a little bit away from the phenotype. There are regulations for protein translation, for protein folding and activity that sometimes does not translate well with phenotype. Proteomics and metabolomics are closer to the phenotype, but they are harder to cover. Proteomics, you can run into membrane proteins, although it is still easier to do for proteomics because you can easily use only, again, one technique. Either do a 2D gel or a shotgun using LC-MS. For metabolomics, it's the hardest because the nature of the metabolites is different. You would see different solubility, different structure, molecular weight, hydrophobicity. There is no one method that will cover a breadth of metabolites as you see in the other 3. So that's put a little bit of a challenge on doing the omics. The metabolomics is usually done by 2 different techniques, either mass spectrometry or NMR. The majority of the data comes from mass spectrometry because it's a high sensitivity and resolution. You can have thousands of signals, usually when you couple it with liquid or gas chromatography, versus the NMR, it's higher throughput, inherently quantitative because you can measure the proton, and that can give you, integrating the proton peak, and can give you the calculations of the a quantitative calculation and assessment without having to use standards. But it's a low resolution and will result only in 30 or 40. So for a crop, assessment, using targeted, we do more. So it's not very appealing. So a lot of studies, rely on the mass spec. So again, this is just a typical workflow. You got a sample, you have to do the extraction in multiple solvents, organic solvents and aqueous solvents. You use the organic solvent for lipids and lipidomics. You use the aqueous solvent. You either do primary metabolites were done mainly by GCMS, the secondary metabolites by LCMS, and the lipidomics also by LCMS. There are multiple mass spec. There are time of flights. There's the mass spectrometer. I don't want to go into details. There are Orbi traps. So there are, even within the same time of flight, there are different vendors. You'll find different softwares between them. Standardization of that also, is something that we keep an eye on and we take into consideration. This is again, kind of like a typical workflow, or at least that's how I used it a lot in Bayer and in my years before joining industry. I was in a research institute. You collect the data. This is the LCMS trace. Under each peak of those, this is the masses. So you end up with having tens of thousands of mass to charge ratio. So you have M plus H, M plus sodium, plus potassium. So all the masses that a software can pick. And then you have 2 workflows. What are you doing? Are you looking for a biomarker, for example? You have, group 1, group 2, you want to see what's the difference between the two. Then you don't really care much about identity. You take all those thousands and you start doing multivariate analysis, PCA, OPLC, and this is an S-plot that you can do. This will highlight the differences between two groups. So there's one group and the group. These are the component. These are the mass signals that are higher in this group and lower on the other, and that vice versa. So these are the components that are mainly responsible for this segregation in the data. This is one group and the two group. And the reason those are those metabolites or those signals are the most one. So you forget about all the rest, and then you can focus on identifying those and validating them and trying to see what is the difference between the two. If you want to have a mode of action story, if you want to see, actually what genes, what pathways have been up, what pathways have been down, then the identity becomes really important. Not only the differences, you want to see a lot what is going on. So in this case, you spend and invest a lot of time trying to identify and annotate the metabolites, and then you can plot them on a pathway map to see what genes have, or what pathways have been deregulated, what pathways have been upregulated. So that's very important when you do like a mode of action study, for example. This system is variable. So as I talked earlier, when we were doing traditional with targeted assay, the experimental design is very important. Here, it's even much more important, is that to have your experiment well designed to block in, reduce any uncontrolled variations. You're using randomization, for example, assign enough experimental unit to your treatment to be able to detect it, to detect important effect, block any unwanted variations. If and since usually it is used in discovery, sometimes this is not very well thought of, at least in the papers that we see sometimes, and it becomes extremely hard to interpret the data. So experimental design is the first step to really get a biological sense of your profiling, whether you're doing transcriptomics or proteomics or whatever, because the variations come that you're not targeting something with the standards and no, you're just looking at everything with one method. So it's not optimized for everything. It's, it is a compromise between all the methods that you want to see. So that's where the variations come from. We do all all our sampling standardized, minimize sampling time, avoid diurnal effect. We never sample, at 9 o'clock in the morning, because if you have 2 hours from 9 to 11, you are at the slope of the diurnal effect. So we usually focus on, 11 to noon, or where the least change in the diurnal effect. Barcoded samples and trace them. You freeze dry or flash freeze them in liquid nitrogen just to maintain their status, and then you transfer them to the lab under dry ice. And that way we can, again, minimize all variations that comes from sampling, mixing samples, or or mislabeling, or all these also steps can really make it difficult at the end for a researcher to look at the data and make sense of it. So from field to lab, everything has to be tracked, and then comes the variations from running the experiment. And I'm sure you are aware, if you're working in metabolomics, about the quality assurance and quality control consortium. If you're not, check them out. There's a lot of publications. There's a lot of best practice to use. Randomize your sample. That's extremely important, because it's going to mediate the systemic bias. You run blanks so you can see whether you have any carryover in your system or not. Use internal standards, because again, your data is not standardized, so you have to use internal standards. These are standards that you're going to add to each of them to have a relative quantitation between your signals. Monitor performance. A pooled study sample is extremely important to run across batches. So if you have a large data set that you are running, and this is an example for it. For example, you run a batch, and then you run another batch and another third batch. And again, this is true for both targeted and assays and omics. For targeted assay, there is a QC sample. If you go to any GLP lab, the first thing you can ask them about, what is your QC sample? How many times you run it in your queue? Once they see this QC samples is dropped, they stop. They they ignore that batch or repeat it. So they have a criteria that the batch has to be within 1 or 2 standard deviation, I think 2 standard deviation of their QC sample. For an omics, you have to have something similar to that. So you have this red lines there or red dots, that's your pooled sample. Once your pooled sample is lower, you can normalize against it. So that's after inter-batch correction or normalization. That can help. And again, you can see the publication there. I think they talk about it, and they have a lot, other techniques. So this is again minimize the variations that you see from that. I just want I'm not gonna get too deep into this, but I want to give an example of a successful story of how we used omics and metabolomics in particular in Bayer. We were trying to identify a biomarker for the moth that was fed on corn, cotton, and soy. We want to see whether this did feed on them, and this is very helpful for us from the insect-resistant management system. Corn is easy to segregate because it is a C4 plant. So there is a technique called isotope ratio mass spectrometry. We measure the 13C to the 12C for the moth wings. And in this case, we can figure out what is the moths that fed on corn or any cereals that are also C4 plant versus 3. But to differentiate between soy and cotton, we had to do an on-target metabolomic. For both, you analyze the plant and you analyze the moth and you see where's the biomarker. In this case, we were looking for biomarkers. We didn't look for we looked for the significant differences in signals between the one that was fed on soy versus the one fed on cotton. And we had hundreds of biomarkers. But the hundreds of biomarkers have to be validated, because I can tell you from experience, many of those biomarkers was false positive. Finally, we were lucky to get a biomarker for cotton. And we had to do the traditional LC, prep LC, NMR, using just databases and isotopic mass and accurate mass, even at 1 ppm or 5 ppm, were not enough for us to identify what's the structure of this compound. So we had to scale up the process. We had to use preparative LC and collect the sample, send it for NMR, and identify that the biomarker. It was just published last year. We weren't even lucky for soy. We had, I would say, tens of potential biomarkers. But every time you repeat the experiment, every time you validate it, now you take it, you validate it with an LCMS, a mass that is targeted for this signal. You're not looking for everything anymore. You identify that signal, this is your biomarker. Now you go in and you develop a method that will only look for this with a higher degree of certainty, and it doesn't hold. And validating biomarkers is also a big topic right now. In FDA, there is a qualification of biomarkers and validation that usually comes up from profiling, whether, again, transcriptomics, proteomics. We have to really validate. If you want to read more about that story, of course, it's much more in details in this publication. We weren't so lucky when we came to yield. So we wanted to get a biomarker, a metabolic biomarker for yield. This would have been a great business benefit for us, because imagine we have hundreds of varieties. Instead of going to the field and spending all the expensive field trials, if we have a method in the lab that we can screen all those and we have markers for yield and we know which of these varieties will have high yield, which would not, that would have been a great benefit for us. But it became elusive because trade is not a simple, or yield is not a simple trade. Yield is a complex trade with multiple trains and genes that affect yield. And we found that the metabolomics is affected more by the environment and the genetic. And that's, again, the same message that we find from one experiment to the other. It is mainly a representation of environmental and genetic effects. So, and the genetic part was much lower. We did not have any inheritance. And again, it's I don't want to go through all the complex figures. If you are more interested, you can see the reference there. But as literature showed that environment and genetic background has much greater impact on the metabolome than the transgene. Again, this paper would show here that we have data from two sites. You can see that the segregation is based mainly on the site first. And then when you look between each site, you will find they segregate based on the variety. Some of those varieties are genetically modified and some not. You don't see the segregation based on these are genetically modified and these are conventional. Most of the segregation, mainly environment, and within each environment, it goes for the genetics. So last year, we kind of like also see a lot of, as you are aware in the literature, there's a lot of call for using omics and using large data sets in risk assessment and using metabolomics for sure. So we set up to write like a review article that actually got a lot of attention. So we published it in October, and it was the second most downloaded review article in Metabolomics in 2020. That we, after surveying what is published in the literature, I guess from 2012 till 2019, we focused on what is needed, what do we need to improve on in metabolomics to be ready for risk assessment. And we identified four major disciplines or four major categories. The first one is tools to interpret the metabolomics data for genetically comparative safety assessment. How are we going to, so we're going to have two big data sets. How are we going to make biological sense out of it? Are we going to compare each M over Z to M over Z? EFSA had a conference in Italy, in Parma, I believe, in 2018, and they discussed the same. And I think we also mentioned that the paper they published in our review article, and they advised against that. They wanted to have a holistic overview of the whole, because you don't want to go M over Z over M over Z, because again of the complexity of M plus one, M plus sodium, adducts. A lot of variability will make it very difficult to compare, signal by signal. So they want to have our endpoint by endpoint, as we call it in traditional. So they want a holistic one. The second, I think category that metabolomics have to get a little bit better on is signal identification. If you are lucky, 50% of your data will be putatively identified, and 50% will be total unknowns. Again, when you're in the biomarker, it's easy because you don't rely on a lot. You just focus on the most differentiated signals between a group, and you can spend then a lot of work trying to identify those. But for if you want a holistic view, then we need to do better on libraries, on databases, on MSMS identification. And I'm not saying that this is a very active area of research in the area of metabolomics. There's always new databases out, new software is out, new algorithm try to identify signals out. It is just needs a little bit more. And then the challenge again with using omics is to capture the diversity. So which method are we going to use? Are we going to use an LC method or GC method or both? And then you have another additional of integrating two different data sets together and normalizing them. What about the components that are not going to be measured by metabolomics, like vitamins, for example? Vitamins are very low, and then again, metabolomics, you have to understand that it will pick the largest signals. Low signal like phytohormones or vitamins, some vitamins, and all this is not going to be picked up. Minerals, you have to do an ICP-MS, a separate method to get them, and then fibers and proximates and all this that's a whole different story that will not be captured by that. So again, in that EFSA conference, this discussed, should we replace the traditional with omics? And the majority voted no. They came to a consensus that metabolomics can be useful if we don't have a mode of action. For example, if you are transforming a transcription factor that you don't know what is it doing, you don't know actually what is the mode of action for the transcription factor, and then something like that would be helpful to give you a hypothesis to build on. So at the end of the meeting, I think there was a consensus to use it on case-by-case basis. They still identified the need for using traditional methods, using targeted methods and assays, but they saw the benefit of using omics as a case-by-case basis in case a mode of action is not clear, for a developed product, for example. And then, the point that everybody agrees on is that standardization of the method is different. Is important. If you run two different vendor softwares, you will get two different results. If you run the vendor software and you run a third party, MZ Cloud, for example, you will get different results. In our lab, we run it usually with minimum two. So we run it with the vendor software. If we're using a water software, we use marker links to find out all the statistical, all the method, peak identification, integration, and all the data. And then we use a third party as well. And we focus on the signals that are common, because you don't want to spend a lot of time in your lab and set methods for false positive. If the both different softwares do not pick the same signal, most likely it is a false positive, or you have to really go in and check the peak integration and to see whether it's manually. You have to do it manually. So again, those are the four main blocks, and there's a lot more to talk about in the review if you interested to know more about that. But from the tool to interpret the data, Dr. Esther Cook from Netherlands, she published a one-class model. She developed a one-class model a few years ago, and they used it for transcriptomics data first, and then they extended to metabolomics. And these are this is, I think, the first publication that they published on it, and then they followed up with two different publications afterward. So basically, you get the profiling of a known safe crop. That's the conventional crop, which is either transcriptomics, metabolomics, and you build a one profile classer. And then you use that profile classer to test a new variety. In this case, if it's a GM variety. And then you have a threshold. If your new variety within the threshold of the safe class, then yes, it's safe and there's no further analysis is needed. If it's outside of that model, outside of that class, then further analysis is required. And I'm not going to go into, in sake of time, and into how this model is built, but you always find it in these references. I will pick an example from this paper that was published a couple of years ago, and it was on corn, on maize, and this maize was MON 810, and it was part of the EU-funded GRACE project. GRACE project was a 3-year project that tested the 90-day rat feeding studies to see any unintended effect. And then when they saw none, they extended to a year. They saw that maybe 90 days is not enough. We will extend the feeding for the rats for a full year. And the conclusion was, again, MON 810 has not induced any adverse effect. It is not, and increasing it to a year did not provide relevant additional information. There were 2 different studies happened on that. Another EU study, we called the G-Twist. Again, that was for 90 days, subchronicle tox, 1 year, 2-year carcinogenity study. They extended from 1 year to 2 years. And there was a French study they called GMO 90+ that looked for metabolic changes for rat, also feeding on, and I believe also MON 810 was part of those 2 studies, and the results from them confirmed that. So and MON 810 is actually one of the most studied and most registered events in the world, in all of the regulatory agencies. So in the paper, the class model that was presented, there was 2 data sets. One data set from Ricklet, this is from Netherlands, and one data set was from the UK. And these are the safe conventional varieties that was used. So total classification, number of classification in, and that's the percentage. So this is the total data set. So, just to simplify things, when you test your GM, it has to have a percentage of higher than 64 to be called in the model, in the safe model. that's from the model that was developed in Netherlands. In the model developed in the UK, you have to get an 80% to be in. But there is limitations. Even see, these are the background. These are the non-GM background of the GM tested. These two were the near isogenic lines. They didn't pass the threshold. So that is a limitation. If I am going to develop a model to say if it's in and out, and my control, the one that I use as the nearest isogenic line, so that tells me that the genetic variations in that line that I'm developing is different. This based on genetic differences between those near isogenic controls and the conventional. And they agreed in the paper that this model failed to assess that. The other model, one failed and one not. So one near isogenic line was within the model, and one near isogenic line was out. So the limitations that I see in this model currently is that two different data sets, two different models, two different thresholds. Even within one of them, even the more successful one, still one of the near isogenic line did not make it. So it is a great tool. It is a great step forward. I think this is the first time has anybody have attempted to develop a tool to describe the metabolic data in risk assessment on building a model such. But I think there is still further development required to have it, because I don't think regulatory agencies, if we run two experiments and we get two different results, one in one lab and one on the other, or one data set and the other, how are we going to interpret this? And this is using a MON 810 that's a safe one, so it's easy for us to save. But if it is really a new, untested, or haven't been, we just newly testing it, we won't have that. We would like to have this security or validity that the model we are testing is right. It's not a false positive or false negative. They used fungal infected samples as a false positive, or like as a control for an outside model, and it was out. But again, if you look at the GM events that both MON 810, one of them okay, here you can say, or by the threshold, one of them okay, which is the yellow one. The other the yellow one is out. I'm sorry, the yellow one is out, and the unmarked one is in. So that's again the threshold from the first model, 64.1. The first GM event was less, so that's out. The second one is higher, so it's in. In the second model, both are 100%. All the subclasses was within so all of them are in. So again, it is the first time we see something, someone developed that, and it's a great tool, but I see limitations in implementing this. There is still more validation required to have this ready for regulatory submissions. Another thing from the paper as well, this is again the S model that I talked about, and these are one of the GM crops and one of the control. This is the isogenic control for it, and they focused on those three metabolites or three signals that differentiate the two. This is the fold change. This is the retention time of the peak and the mass to charge ratio for it. And that's the molecular formula that you can deduce from the isotopic distribution of the mass signal and the mass and the accurate mass. And this is the Kim spider structure. I guarantee you that these have to be validated. They haven't done any validation, but just look at this one. That one has C13 in it. This is the top score for searching that mass and that molecular formula in Kim spider, and it came up with a compound that has a C13. This is not available in nature. In corn, does not have a compound that is enriched. This is a synthetic compound that's just matter, and this is very common. And they say that in the paper. They don't, they say that one of the identification. I'm just putting this as an example of how hard it is to identify. That's why we could not identify our biomarker that we got from cotton using Kim spider and databases alone. We had to really go through the LCMS and the prep LC and get it and do MSMS and do NMR and do a lot of other traditional chemical identification, like a phytochemistry study, to try to identify the structure, because it is extremely difficult. And if you have an isobaric compound that compounds that have all the mass, the same mass to charge, they are just structurally isomers. Now it's becomes even more complicated to try to identify using MS and mass, but it is, that's what I'm trying to say, it's challenging to do. I am out of time, but I don't want to go too much in. This is just as an overview that the way we look into untargeted metabolomics is great for discovery, building hypotheses, versus when you use validated is great for regulatory and hypothesis driven. We know what's the hypothesis. We know what the mode of action for the gene that we entered in. So you can have an absolute quantitation using authentic standard, using confirmed compound identification, and reproducible as validated. These area still needs to be more development for them to use. So as a conclusion, and I'm sorry going 5 minutes over, GM crops are extensively tested for food and feed safety. There has never been an adverse outcome of GM crop. Target composition are more suited and adequate to assess GM crops through regulatory process. Metabolomics is a great discovery technique to generate hypotheses and to generate biomarkers that needs to be validated. The biomarkers have to be validated with and quantified. And from our opinion, is further developments are required before implementing it successfully in GM crop assessment. Thank you, and I apologize if I was speaking a little bit fast, but I was trying to cover a lot of information in the time. And I'm happy to have any questions.
Thank you so much. I would like to thank you for your presentation. I think your presentation was very full about GM food crops and metabolomics. And I saw if you use the omics technologies are of increased importance in research. However, analysis of omics data is complex. There is an urgent need for the standardization of analysis workflow and the reporting of study findings. Thank you so much. I don't know, is there any question from our audience?
I have the first question. I can go. Okay. The first one is about CRISPR. It asks, CRISPR-Cas system is a powerful tool to upgrade and breeding in crop. It asks, what is your opinion about CRISPR-Cas system and how are you using this tool as a buyer? From Elif.
Sure. For CRISPR, we are right now using it as a tool to develop. Some regional, like the EU, still consider it a GM, so it has to go through all the GM regulatory agency submission and regulations. Other, agencies like in the FDA, it depends on what the type you're going to use. If you have a single mutation, if you have multiple, so it is an active research that we have it in bio crop science. So it is an active in biotech. We don't have anything in regulatory based on gene editing yet, but that's an active area of research that we are pursuing. And not only Bayer, I think other companies as well are using gene editing and the CRISPR system to develop their products.
Mm-hmm. Amazing, thank you. And the second one is about metabolomic, and it says metabolomic has developed into a major tool in plant system biology, and you explained the impact of experimental design is particularly important in metabolomic studies. Well, what should we pay attention to, especially in the extraction step, in order to get meaningful result? But generally only one extraction method is used, so I would like to ask that from.
That's a great question. The method that you're going to use for extraction is the first area of differentiation you're going to see. If you add an acidic, then if you want to, for example, hydrolyze all the glycans or anything, or you don't want to do that, what type of solvent? If you add chloroform methanol water, is going to be a little bit different than adding, isopropyl. And there are multiple methods. You just have to understand that variability can come from this. In our, you can't have one thing that will get all, but what we do is we use a system that has both an organic and aqueous, and then we separate those the two. The organic, you can use it as is, or you can hydrolyze all the fatty acids into fatty acid methyl esters. So this is a very critical step to standardize in your method and to make sure that the method is applied, especially if you have a large data set. When you run, we used plate-based assays, 6 well plates. You can use also vials. It depends on what your batch is. Standardization, adding your internal standard yearly, making sure that you have maybe more than one internal standard that covers the organic phase part of your analysis, the aqueous phase part of your analysis. Just be aware of that. Be aware that this is a critical step that can cause problems later on in trying to understand. And then all the other standardization that this paper that was published in Metabolomics on the standardization is very important, and provides a lot of guidelines on how to normalize, how to use proper internal standard controls, randomize your sample, randomize your analysis when it comes to your LCMS or GCMS. That's very critical.
Okay, maybe I got a question from Sena Çakır. There is another question. Maybe this one is last question for you. There is a hesitation to use GM crops in Turkey. That's maybe because of the lack of knowledge of people, or maybe information around this topic is uneducated to convince people to consume GM crops. We can see safety assessment of GM products are took seriously, but as the experts on the subject, I want to know what is your approach to the possibility of a mechanism that unnoticed yet and can cause problems in the future.
I'm sorry, I missed the last part. Can you repeat the last?
I want to know what is your approach to the possibility of a mechanism that unnoticed yet and can cause problem in the future.
If a product that's not well developed, it's not well tested, then you can have an unintended effect that you don't test. You can have an effect on on environmental effect. If you have a product that was not tested on non-targeted organism, for example, that if you have an insect protein that is not, again, well tested, and you're targeting a specific pest, but other non-target organism can be affected with it, that will have an environmental effect on an assessment on GM crops. The key, as I said, is the well studied and all the work that is done. And I know from industry, when I talk about that from the industry, sometimes they would say that you're just talking because you want to promote your products, but safety from industry is critical, because the implications of putting a market of product out in the market that has a safety issue or has an unintended effect on environment is business. It has a huge implication on business. So no biotech company would like to be in that position. That's why we test and we test and we test, and we always select the best product that we would have the safety data to stand behind. And that's, again, the responsibility for the regulatory agencies, the responsibilities of the academic research and universities to take this and test it further and to verify that. I think the problem that comes from the GM and the use of the GM is not based on science, rather based on public opinion. There has been a public opinion that this is unnatural. It causes and it must not be good. It's not naturally. So when you talk to EFSA, when you talk to other reagent, if you talk from the science and you provide the science that prove that, they cannot dispute it. The problem that comes from sometimes the public opinion on genetically modified crops. So I think there is a lot of work that needs to be done educating the public about the safety, about the studies that are outside there. And I think in Bayer we have a program that we will put all our regulatory studies public, so everybody can see it, everybody can review it. So there is and I think that's a trend that's going also through other biotech companies, transparency and making the data available for academia to review and to assess the science behind it.
Yeah, thank you so much. We are getting so many questions for you, but there is no enough time for all questions right now. Maybe I hope you will want to be involved in our meeting next time, and I hope we can do next time face to face in here in Turkey.
I hope so.
I'm very pleased to have your participation in the Biotalks. And I thank you again, Bilge.
Thank you again, Dr. Bedair.
Thank you very much. Appreciate it.
It
Thank
was
you.
really interesting and inspiring.
Thank you.
Now it is suitable for everyone. I would like to go on to